Thank you to everyone for the interest in my presentation and for Toshiba Medical for inviting me to this wonderful symposium here, which I personally very much enjoy as a basic scientist somewhat, though in the neuroimaging field, but I don't have a formal training in clinical research, or in clinical applications actually, so this symposium is really interesting for me. If you look at the slide, I'm not an associate professor, and actually there's another error in the program that I want to point out. I don't have 200 publications yet, because then I would really be tenured already. So I would love to have 200 publications. Okay, so this is about advanced susceptibility MRI and my primary research field. So the presentation will be about quantitative mapping of a physical property. So before I start, I want to talk a little bit about our lab. We're just 20 minutes from here in Buffalo, on the Buffalo Niagara Medical Campus, and few people actually know that the buffalo wings were actually invented on this campus, basically, in the Anchor Bar, and right across the street is the Clinical and Translational Science Institute and the Gates Vascular Institute. So it's a building co-owned, pretty unique building, co-owned by a hospital provider, Kaleida Health, and the university in Buffalo. So hospital first 4 floors and research on the upper 4 floors. So the connection is made by the Jacobs Institute, which brings in companies and facilitates basically the application of new technology in the clinical setting. So the surgery units are right on the 4th floor, and there's a stairwell that connects these 2 labs. Then we have on the 8th floor the world-renowned Toshiba Stroke Center, where they develop new angiography detectors, for example. And in the middle, we have the Clinical and Translational Science Institute and my lab, the MRI Biophysics Lab of the Buffalo Neuroimaging Analysis Center. So in the CTSI, we have a few scanners. One of them is the Toshiba Vantage Titan 3T scanner. This is a research-only instrument. All these scanners are research-only, so there's no clinical scanning there. And we are very grateful to have this machine, which is actually provided through a collaboration agreement by Toshiba. So that's basically my disclaimer, my conflict of interest here, if you will. Apart from that, I have nothing to declare. We have a 9.4 Tesla scanner with a cryogenic coil, which allows us to do excellent research in mice and in rats, and a PET-CT scanner, and then there are a couple more Toshiba MRIs and GE MRIs that are in clinical use in the hospital downstairs. So with respect to the 3T MRI, we're trying to start some clinical studies there, clinical trials. Right now we're performing preclinical studies. That is primarily pig studies, cardiac research, and stroke research in dogs. And we do methodological development that's primarily accelerated imaging with compressed sensing. We do microstructure MRI, and what my research interest is really is quantitative MRI advanced techniques. So one of these techniques that we are currently exploring is, for example, megapress. That's a technique, as edited spectroscopy technique that allows us to measure neurotransmitter concentrations, GABA and glutamate in the brain. We have a functional perfusion mapping study, diffusion tractography, and the method that I'm talking about today is quantitative susceptibility mapping. And I have to say that we moved basically from our GE platform to this Toshiba platform, and Toshiba was incredibly supportive in bringing these advanced protocols to this different platform, which is not only in a clinical setting a problem, but especially in a research setting. So why would you do quantitative MRI of a physical property? So this image shows you on the left side a healthy brain, and on the right side severe Alzheimer's diseased brain. And MRI is pretty good, a pretty mature diagnostic imaging modality to show this structural change on the images, but in the end it's just shades of gray oftentimes. MRI is not really an excellent measurement instrument, so we just get shades of gray. And what does that mean? Actually, these structural anatomical changes that we see on MRI, they are often just a very late stage of the disease. So we can see them nicely, we can do a quantitative analysis. For example, many of our clinical trials, we assess the brain volume as an outcome measure, and for example, to see a drug efficacy, but it's very, very late stage. So what we are actually interested in the research setting, and we also want to bring this to the clinical setting, is to assess the change of the biophysical tissue properties even before we see a structural change. And I want to show this here. This is a nice landscape colorful image. This is what we call an image. It's a qualitative representation of the reality. And what you can see is there's a boat and there's a mountain and a lake, and you can now do some quantitative analysis on that. For example, you can say, oh, okay, the mountain is 1.5 times as high as the boat. And this is correct in the framework of this image, but it does not represent the physical reality. So where we want to go with MRI, and that's maybe an outlook to where MRI will go in the next 10 years, is quantitative MRI. We want some specific numbers. We want that the image intensities in the images don't depend on the sequence parameters. They are actually physical properties independent of the scanner and tell us something about the physical reality. So we believe that with this quantitative imaging approach, we will be able to do a quantitative analysis again, and then be able to elucidate disease mechanisms, have a biomarker for drug development, and also in the clinics, we'll be able to define clear treatment cutoffs, for example, or monitor disease progression. So that's the overall motivation. I'm particularly interested in brain iron, basically, since I started my PhD thesis. So if you look at these post-mortem slices of the human brain, you know, if you're not familiar with them, you notice that there's blue is a Prussian blue staining, so blue means there's iron. So you see some iron in the cortex, but you see this peculiar pattern of iron accumulation in the deep gray matter, in the basal ganglia in particular. So this is well known that there's a lot of iron, but we really have no good idea what this iron is about. Why is there so much iron in these regions? So we know that iron is a vital contributor to brain function. For example, blood transports, the heme molecule transports the oxygen. Cellular respiration requires iron. Myelination in the oligodendrocytes requires iron. But if we do an estimation, only 5 to 10% of the iron in the brain are actually required for these physiological processes. So what about the rest of the 95%? And one thing that you have to consider when you talk about these high concentrations of iron, they are as high as they are in the liver, actually, in the basal ganglia, that iron is a very active oxidizer. So iron triggers inflammation through the promotion of the creation of hydroxyl radicals, for example, reactive oxygen specimens. And that has been implicated with neurodegeneration. So we are very interested to explore the role of iron in aging and neurodegeneration. So this is one of the first papers where it was noted that iron might have something to do with neurodegeneration and with neurological diseases, from Burton Dreyer, 1986. And the images on the right that you so here, what you see here is basically a T2-weighted image from a long time ago. And you see that the globus pallidus here is significantly darker than in the healthy control. And that was basically one of the first indications that there is iron accumulation in multiple sclerosis. So we were thinking, so how, if we are interested to understand the role of iron, this is very difficult because iron changes very slowly over decades in the brain, very difficult to study. How can we measure iron in vivo? So iron, if you are an MRI person, you know the sign, the magnet is always on, so it has something to do with magnet. MRI is a magnetic field. And then you remember iron, you should never bring iron into the magnet room because it gets attracted by the magnet. And this attraction is because iron becomes itself a little magnet when you expose it to a magnetic field. And this property is described by the magnetic susceptibility. So that's a physical, basically a number that describes how strong the material magnetizes. And that property is not restricted to iron. Actually, every biological tissue, every substance, water, glass, everything magnetizes when you bring it into a magnetic field. So a side effect of that magnetization is that the overall magnetic field is also not anymore homogeneous. It is also perturbed. Perturbed. So radiologists, oftentimes when you mention susceptibility, they first think about susceptibility artifacts. So if someone has an implant, you get these ugly artifacts, you cannot really interpret the scans. So a little bit more and more coming is a technique called quote susceptibility weighted imaging, or T2 star weighted imaging, and that's a technique that actually uses the subtle differences between different tissue properties, the subtle differences in susceptibility, to create a contrast. And what you can see here is, this is actually 9.4 Tesla human image. You can see extremely small vessels in the human brain, veins, and this is because deoxygenated blood magnetizes slightly more than arterial blood. So this is why you see on these SWI images, you see primarily veins. And it's also an excellent technique to see microbleeds, for example. So this susceptibility-weighted imaging technique, or T2 star, relies on this typical MRI image. So if you see an image like this, it should look familiar to you, T2 star weighted image. All MRI contrasts look more or less similar compared to this image. And this image is a face image. So SWI, the susceptibility-weighted technique, actually behind the scenes uses this data, uses this data to create this nice vessel contrast. And you as a radiologist, you never get to see this image. But interestingly, every MRI sequence gives you this additional data that is usually just discarded. It's just thrown away. The scanners don't reconstruct that. So, but it's interesting, if you look here, you see actually there's considerable amount of information already on this face image. So what we are now doing is, and this face image represents the magnetic field perturbation. So what we are doing is we take this image that's full of artifacts, or apparently looking artifacts. We perform a mathematical technique called unwrapping to eliminate these lines of black and white lines here that are called phase wraps to get a quantitative map of the magnetic field inside the brain. Then the next step is that we solve, so what you see here is these black and white regions. They are basically large-scale magnetic field perturbations from the lungs, from the sinus cavities. And we now solve a physically motivated differential equation, Laplace equation, Poisson equations, to extract the field that comes only from the susceptibility distribution inside the brain. So this is the field perturbation of the brain tissue itself. And then once we have this field perturbation, we can solve another mathematical three-dimensional problem, which is a convolution integral problem, to calculate the magnetic susceptibility distribution that creates this field perturbation. And these are exemplary image here. This is a 7 Tesla image acquired in Germany. What you can see is that there's excellent contrast between gray and white matter. You see the veins here. That's maybe the reason why I'm in the vascular session. And you can see the iron-laden basal ganglia regions. Really, really nice and very unique contrast, very highly sensitive, more sensitive than anything else we have for iron. And we also, in a post-mortem study in deceased subjects, we scanned them in vivo with this technique and then extracted the brain, not in vivo, sorry, in situ, so the brains were still in the body, in the skull. We extracted the brains, we measured with inductively coupled plasma mass spectrometry the iron concentration, and we found a pretty linear concentration, so the susceptibility of the brain is in fact dominated by the iron. So this is one of the hallmark papers of iron in the brain from 1958, Hallgren and Surrender. So they studied about 100 subjects over the whole range of ages from 0 to 100 and found these exponential curves. So that's something that's known. Iron accumulates over, like, etc. It's somewhat 30, 40 years. But what you see here is there's a considerable variation between individual subjects. So if you want to use such a technique for research or for even for clinical cutoff values, for example, you want to diagnose a disease, you need to understand what's the difference or how you explain these differences between individual subjects. So to do this, we apply a pretty interesting, currently employing a pretty interesting mathematical approach. So what we do is we try to describe every single map of a patient by a linear combination, so a weighted combination of certain modes of iron accumulation in the brain. Say, left and right, globus pallidus accumulate iron always together, the putamen, red nucleus, and when you add this all up, you get the patient's iron map, if you will. So once we have these characteristic modes that describe the iron accumulation in the brain, you can actually describe every single subject with just a couple of numbers that describe the combination of these modes. And so the question is, how do we get these modes? And that's actually a blind source separation problem. So we apply a transformation called independent component analysis to about 250 healthy controls. And then we get these modes, and we get the coefficients. And this is, for example, one of the modes here that describes the variation between individual healthy adults. And what you can see is that the red nucleus and some part of the putamen always accumulate iron together. And there's also some caudate and so on. And all these components look slightly different. So I'm showing this here. This is one of the components, actually maybe the one that was just rotating at this maximum intensity projection. What you can see here is a correlation over age. So you see there's a pretty good correlation with age. But there are other components, and other components maybe have some kind of a nonlinear relation or have no correlation with aging. So that's pretty interesting. So you have variations between subjects that are really not related to age. And then the question is, of course, what is this caused by? Is this environmental factors, or is this some disease process, or is this some accelerated aging, or something like this? So this is what we are currently exploring. And then the next step is to interpret these modes, basically. So why is globus pallidus at the same mode as the putamen or the caudate, for example? And we believe that this is somehow related to the structural connectivity and the functional interactions between these regions, so that potentially with this decomposition we are able to see functional differences and structural differences with a completely different technique than that is currently used, like DTI or fMRI, or in combination. So, for example, you know that the nigrostriatal pathway is affected by Parkinson's disease, and for example, Huntington's disease affects like this indirect pathway here. So it might as well be that when you look at the susceptibility maps of these diseases, you can identify actually different modes of iron accumulation. So this is shown here. So we then represent this as such a correlation matrix, and we believe that these different diseases have different network signatures in the iron concentration. And this is some preliminary data, a little bit simpler analysis than what I've showed you just a couple of minutes ago. So this is a fairly big study with more than 1,000 MS patients where we applied this technique. And we indeed found that there's a significant accumulation of iron in the caudate, in the putamen, and globus pallidus. So that's pretty consistent with these very old qualitative results. But we now have a much more sensitive technique. And we actually found that there's a significant decrease in iron in the thalamus, decrease in magnetic susceptibility. So that's completely the opposite of what's a common notion about iron in neurodegenerative diseases. So we looked a little bit more carefully in subgroups of multiple sclerosis, clinically isolated syndrome, relapsing remitting MS, and secondary progressive MS, and with a technique that uses a deformable model so that we can actually look on a voxel-based level at the susceptibility changes. And if we do this in the thalamus, you see that the decrease, so blue means decreased iron concentration compared to controls. What you see here is that actually it's restricted to some nuclei of the thalamus. So what is important to understand is that the nuclei of the thalamus, they are connected, they have distinct connections with cortical regions. So we believe that the cortical damage that occurs in multiple sclerosis actually comes first and then propagates along structural connections to the thalamus, where the inflammation results in decrease of iron, and that can actually be explained by for example, cytokines that release iron from oligodendrocytes. So, this is basically the story around the iron and this novel technique that, as Guy mentioned, may actually be available on the Toshiba's platform, at least as a WIP package sometime down the road. So that's actually, I think, the first vendor that really presents it this way. Some other vendors are also working on that, but not on a direct integration, I think. So. This is iron. So iron actually, what iron does is it increases the magnetic field in the iron-laden region. But there are also substances that actually do the opposite. So they have a negative compared to a positive susceptibility. They push the magnetic field lines outside of the tissue. And that is calcium, primarily in the brain. So if we apply the susceptibility mapping technique to a patient with a calcification, we are now actually, for the first time with MRI, we are possible to distinguish between iron blood, for example, bleeding, or in this case here, just the iron load globus pallidus in the veins, and calcification here in tuberous sclerosis. And we applied this in a study in glioblastoma patients before treatment, and we were able to see, to identify an oligodendroglial component just based on the very specific identification of calcifications on MRI in this component. And so many people then say, okay, you can also see that on CT, but actually our experience is that you miss many calcifications on CT, especially smaller and more diffuse distributed calcium. And this is where I'm coming now, diffusely distributed calcium. So this is a little bit outlook to our preclinical studies. We have a program where we are interested in better understanding CTE, so chronic traumatic encephalopathy. And we study a rat model of multiple mild traumatic brain injury. And this here is an image from an older paper where they showed that there's calcium influx. When you have a rat model of TBI, there's calcium influx into the thalamus 14 days after the injury. And this is a pretty invasive technique, so basically you inject radioactive calcium, you slice up the brain, you use a radio camera, and you measure the calcium. So definitely nothing that you can apply in the clinical setting. So while we were thinking, if we can see calcium pretty significantly with this technique, we might as well see this calcium here. And this is a result that we are about to publish now. So this is an axial view for those of you not familiar with rat images. Here's the white matter, and this is the thalamus. So you have some hemorrhage here from the traumatic brain injury, and you see clearly the calcification is hypointense here. And what is really interesting is that if we do histology here, that it really exactly resembles our MRI findings. So if you're able to see such a calcification in a brain that small compared to the human brain, then we were thinking, okay, we should definitely look into clinical cases and to humans. And so these are the results that I'm showing you now. This is basically the last topic I'm covering. Is a retrospective study. So at my institution, there were really smart people before I came, and they stored a lot of the raw data compared to just the data that was that the scanner reconstructs. And the reason behind that is actually pretty sad. Most vendors don't allow you to reconstruct face images yourself based on the scanner software, or they are not correct, so you cannot use them for a quantitative analysis. And this was the case here too. It was not a Toshiba scanner. The Toshiba scanner actually produces really nice face images. And I'm not paid to say that, but that's really something we started appreciating. So they basically download the raw case space for all these sequences and reconstructed the face images so that they could reconstruct images in general and SWI images at that time retrospectively. So we now could apply retrospectively this quantitative susceptibility mapping technique. And this is a fairly small study, but we had professional athletes, retired professional athletes, 22 contact sports. I think that was hockey and football. We had 20 non-contact sports, that was skiing, running, cycling, stuff like that. And 25 normal subjects, normal controls that stated that in a questionnaire that they had never in their life played any contact sports. And when we looked at calcifications in the thalamus, and we focused only on the thalamus after these preclinical results, we did not find any calcifications, anything that looked like calcifications in the normal controls. Though we found 2 calcifications in the non-contact sports athletes, and we found 5 calcifications in the contact sports athletes. And this is really exciting, and we're now carrying out a prospective study where we try to confirm this in acute cases. What is really interesting is that it's impossible to see these calcifications on conventional MRI. So susceptibility mapping gives us really some additional benefit here and for this potential biomarker. So if you look, this is even an SWI image that's basically sold as specifically sensitive to calcium and iron and myelin and iron. So you cannot see really anything here. There's a vessel, but you don't see this nice calcification here. This is only visible on the QSM. Yeah, and that's the end of my presentation. And I really have to thank everyone on this slide, all my collaborators that work at the CTSI. And I also want to thank the team in Cleveland from Toshiba Medical. They really help us. We get an incredible support with the sequence development environment, RSDE for pulse sequence implementation, all aspects related to post-processing. So that's something I've never experienced before with the other vendors that we worked with, and I'm really grateful for that. Thank you.